I'm impressed. Okay, while we were waiting for everybody to join in, I did ask if there were questions. Let me offer that again and see if there's something. I saw there was something in chat, but a few things in chat. See if there's anything here.
Chat is just thanks.
Okay, well, thanks are always appreciated. Okay,
That's
great.
right.
Wonderful. And I'm not going to, depends, I guess, on the volume of the response, but Claire had given me a notebook or a model actually that she had a little bit of a problem with. If there are others who have models they're working on and have a little problem, I'm happy to try and respond. I guess if I get overwhelmed, I probably my ability to respond will be limited, but my guess is it's, if there's something you're struggling with, I'm happy to provide some feedback. Okay, well, today we sort of have a double feature. So we're going to do two topics that are, are related, but sort of fill in some of the gaps in terms of doing computational studies. So the first part is, and we've alluded to this on many occasions, is that when you're developing a model, even if it's not a stochastic model, it's a deterministic model using ordinary differential equations, the data that you have to estimate the parameter values almost certainly are stochastic. They're taken from experiments. There's some variability associated with it. So when you construct your parameter estimates using the parameter estimation strategies that we talked about on the second day, it's to be expected that there should be some variance around those parameter estimates and also a variance around the quality of the model itself. And that's what this section is about. There are a variety of different strategies that you can use, and there are a few different parts of this problem. I'm going to focus on really two aspects primarily. The first one is what is your measure of overall model quality? And I'll just give you sort of a brief summary statement. We've been looking at the chi-square value in the output that we get from parameter estimation, but there are a couple of others that are very useful as well. And the second part is how do we go about estimating the quality of the model itself? I mean, you can get, typically the way this would be done is that, or the way you might think of this is that you have a set of experimental data that you've collected. You set aside some that's going to be your evaluation data. You do your fits, and then you see how well your fits predict the data that you set aside. There are a few challenges with this. One of the first challenges is that for most biological experiments, at least at this point in time, you don't have a lot of data. And so, as we've seen before, the parameter fitting itself really requires a significant amount of data, especially for time courses. We want to capture dynamics over time. And if you miss out on some of that data in your parameter estimation, you probably are not going to get good estimates of the parameters. And also the quality of what you're going to see in terms of the testing part of the data. That's going to be challenging as well because some of those key dynamics may not have been included in your training. So it's a bit of a quandary. That's one part of this. And as well as the measure. So those are the two aspects I'm going to deal with. This, I've structured this lecture like I've done with the others. There's sort of a theory part, which should be accessible to everybody in the audience. You'll get the core concepts, so you have an idea about what it is that needs to be done with modeling. And then there's going to be some more technical details that we'll go through in a Jupyter notebook or Colab notebook. That we'll discuss this in a bit more context. So let me go ahead and start with this part just as a forward pointer. The second half of this morning's lecture is going to be about design of experiments. It's a related but very distinctive topic. It's about how do you answer how do you formulate your computational studies to answer a science question? Okay, so let's go on with this, with the uncertainty. So this is the first part about metrics for evaluating quality of your model. There are a few, and I assume that many of you have encountered these before. The first one, which is what's called a non-parametric metric, in that it doesn't require really any knowledge, prior knowledge of distributions, and so it has that generality associated with it, is R squared. And I'm sure you've encountered this before. The idea is that you have residuals, that's the E over here, you look at the variance of the observation. So this would be from your experimental data that you've collected, and the residuals, of course, are the experimental data when you take away the fits. That's the y hat. And this ratio here of variances basically shows how much error or uncertain or unknown, what you are not modeling, is that ratio. And 1 minus this then would end up being a figure of merit. It'd be basically how much you fit. This measure would normally, if you're doing a linear regression, this measure will be a number between 0 and 1. For nonlinear systems, more complicated systems, you can actually come up with negative numbers. I frequently do, just because of the nature of some of the assumptions that are going on underneath. But generally speaking, what you're looking for is something that is in the high positive range, closer to 1. So 0.95 would be awesome. A lot of times I find that I struggle to get things up to like a 0.6 or a 0.7. But it gives you sort of a general feeling. For those of you who have used R squared before, you're also familiar with, it can say that something is good when in fact you have some very strange things going on under the covers. You can have a lot of data at one point and one outlier, and it can falsely tell you that things are good. And that's why I always recommend looking at the data. So another one is the chi-square measure. We've seen this in the reports before. Again, we're looking at the residuals. Essentially, the residuals are being normalized over here. The chi-square, if there's certain statistical distribution assumptions about these residuals, if they're normally distributed, then you actually have you can estimate the distribution of the chi-square and come up with a statistical test as to how good your model is relative to something that was just by chance. The distributional assumptions are hard to verify, and I would be suspect if they often held, but the actual numerical values are still worthwhile in terms of this normalization factor over here, the n minus k, is good because k is the number of parameters, n is the number of measurements that you have. When we talked about before about overfitting, overfitting means that k is really high relative to n. So you sort of expect that you should not be getting as large of a good number if you have a lot more parameters than you have amount of data. So I think that part is certainly very helpful. Oops, I didn't mean to do that. Sorry about that. Okay. And AIC is yet another one of these kinds of more, I would say, statistics heavy or distribution heavy kind of assumptions. You have a factor in here, this L hat, that's the probability of getting the data for the model parameters. A question about where that comes from. But again, you see the K in here as well, which means that there's some adjustment for the number of parameters. So you can use, and all these three and sometimes multiples of them are useful for evaluating the models. With all of them, you're looking for larger numbers, and so you can at least get relative comparisons. And you get the normalizations over here in terms of probabilities. That gives you some frame of reference as to how accurate, how good you're doing. And R squared is just the scale, basically 0 to 1. Although I'll tell you, with bad nonlinear models, you can easily get negative numbers. Okay, so
a quick question
gives
about,
you sort of,
just
sure,
define K and N again real quick.
Oh,
And
sure.
and what
K is the number of parameters and N is the number of observations. And so the intuition here is that you don't want to have more parameters than you have observations, because then you have this, you could basically do anything. It goes back to the von Neumann quote about, with enough parameters, I can fit anything. And that's the intuition. Other
and what does
questions?
AIC stand for?
I don't actually remember. Does
I
someone know?
Yeah, Ikaike. He's a Japanese scientist, so it's named after him.
Okay, thank you. Yeah,
Yeah.
I didn't actually remember. Other questions? Okay, I mean, well, I tend to use R squared because it's simple and I understand the scale, but I don't trust it in the sense that I will always go back and look at the data to make sure that I'm not getting something that just, that a lot of data is clumped in one narrow area and we have a few outliers that are really driving the, what's considered a good fit. That tends to be my sort of philosophy. The other reason is I don't believe we have prior distributions or that we can assume independence or that we can assume normality. So I tend not to trust a distributional assumption there. And that's, again, my bias on that. All right, so that's sort of my take on this.
One other quick question about this. var E, is it var E or var is the absolute value of E?
Oh, it's the variance of E.
Variance.
Yeah, it's actually variance of E, right? Yeah,
Okay.
so you're talking about the how the intuition over here is how for R squared is how much of the variance is accounted for by the fitted values. And so under the, it's more of a linearity assumption where this actually works, is that this is one way of calculating, because the variance of the fitted values plus the variance of the residual should equal the variance of the observations. Okay, other questions? All right, so let me go on to cross-validation. So those are the measures. Then the next question is, well, how do we conduct experiments so we come up with reasonable kinds of quantifications or reasonable conditions under which we're going to assess the data of the quality of the model? And I mentioned before this set-aside approach, where you take data, you've got your total set of data, which again is probably not large. You may have, I don't know, time course, 20 observations. That's actually potentially a lot, maybe more like 10. Although, the 10 time points, although obviously you may be looking at many things. If you're looking at expression data, you could be looking at thousands of genes, but, they're just 10 time points. So, taking out, even one time point could be significant. So cross-validation is a technique to get around that. And it's computationally intensive. So this really didn't become popular until, within a couple of decades ago when, computing was much more widely available. But it's, given that we have, for us, just our desktops, this capability, it's a very powerful technique. So here's the core idea is here's your data, and this little thing up here, this is supposed to be a header, to like label your columns, but the actual data is over here. So, I guess actually maybe I am counting that because I know I wanted to come up. I have, I have 6 rows. I apologize. I guess, that was a little bit deceptive to me. There are actually 6 rows of data. So this thing that looks like a header is actually a data row as well. And the question is, how am I going to use my 6 rows of data to come up with a quality measure? So the idea is you divide the whole data set up into sections called folds. So here's these colors indicate the folds. Now I showed the folds as being divided up sort of like in sequence over here. The technique itself does not specify how it's divided, and that's actually a sort of a design parameter that you have to consider when you're doing this. If you have some machine learning background, you've probably encountered cross-validation already. And in machine learning, the folds that tend to be selected randomly. In other words, blue would be, randomly, one of these, maybe one of those, and red would be one of these, one of those, something like that. But for purposes of describing the technique, I've done it this way. And we'll get back to this in a second, because actually for analyzing time course data, it's very important to think about how you construct your folds. So a fold, again, is a data row. And when you design or a selection of data rows, and then you are then determining how it is you select them, how you decide which folds you're going to choose. So a fold would be a collection of rows. So we have a green fold, which are these two, red fold, these two, and this dark blue one over there. All right, so that's the first step. Then the next core concept here is this notion of training and testing. So the training is what you're going to use to construct your parameter estimates. And if you remember back to our discussion on the second day, what we were doing is we were using the observational data to then try and find the values of parameters that best fit the observational data. But we're going to do that with the training data. So for example, if these two rows here are the fold that I'm examining, those will be my set aside. That's going to be my test data. And I'm going to use these two down here as my training data. So I guess here we go over, I guess actually over here I did it with red. Okay, so red is going to be my test data. I'm going to set that aside. And in this fold, the fold is defined by, I have these two test data and these four training data. So I'm going to use these four training data to construct my parameter estimates. I'm going to apply SB Stoke, for example, to come up with parameter estimates. Okay, and then in this fold over here, so now my fold consists of these two rows here are test, and these four over here are train, and then so on down here. And you see that if you have n folds, you're going to have n pairs of training and test data. So each fold gets a chance to be test. All right, so I've constructed my training and test data. Now I'm going to do the parameter estimates. So that's parameter estimates are all done on the test, on the training data. Okay, and then what I'm going to do is, after I've got my parameter estimate, so let me explain the notation. The theta is intended to indicate the set of parameters. So, we may have 2 parameters, we may have 20 parameters, however many parameters there are. The number in the square brackets reflects the estimates that were constructed from that fold. So these estimates for the thetas were constructed with the dark blue and the green. And these estimates, number 2, were constructed from the dark blue and the red, and so on down here. Then what I can do is, after I've got these parameter estimates, I can then use those parameters to predict the values in the test set. So now I've got a prediction. So now I'm going to have the observed values, which are the red, and then the predicted value from my what I've done over here with these from these parameter estimates over here. And I've shown that I'm going to then come up with some sort of a quality measure. I'm using R squared. So I'm going to have an R squared here for fold 1, an R squared for fold 2, an R squared for fold 3. I'll also have parameter estimates from each one of the folds. And then I can use that then to assess, the statistics associated with the model. So I could compute like a variance of the parameter estimates based on, I have 3 estimates of the parameters. And of course, I have 3 estimates of the overall model quality as well. So is that, before I go on to the more detailed discussion, let me see if there are questions on this.
Also, just like how mechanically would you split things up? Like, is this something you do by hand, something you do by program? And the answer is basically whatever. That's your choice. That's your design.
It's your choice. I mean, ultimately, you want to be able to, and it doesn't really matter how it's done, whether it's mechanically or by hand, in concept. But on the next slide, there's some details related to time course data, and then I'll get into. So hold that one for just a second. Any other questions?
Is theta hat an array of estimated parameters?
Yes, exactly. You got it. And so there, and you see that then there are theta hat, there are 3 different theta hats because I have 3 different training sets. Other questions? Okay, good. Let me go on to the next slide where okay, and then, you could come up with an average value for R squared, and similarly, you could come up with averages for the theta hats. All right, so the folds, choose wisely. I don't know, this comes from, I think it was Indiana Jones and the Temple of Doom, or maybe, I've forgotten what it was, or maybe it was the one with the child. At any rate, there's a scene where
Last Crusade, yes.
Last Crusade, thank you.
There you go.
Right, there's a scene where you want to, you either get eternal life or death if you drink from the wrong chalice. So there are literally in this room hundreds of chalices, and so choose wisely, right? So same thing, you can choose any one of the folds in many ways, but choose wisely. All right, so here's the kind of data that we get. Here's that model, that linear pathway with 3 species. And we see, there are a lot of, even for this simple model here, and I probably should have had much more points. That's why it's a little bit bumpy in through here. So I apologize for the lack of smoothness. But, the dynamics are quite different. I mean, this early
It's your
part
real data.
here,
It's going to be bumpy like that, right?
yeah, and it's going to even be bumpier, right?
Right.
And,
So.
yeah, exactly. So, I mean, in that sense, maybe it's, I probably should have even shown real data. So in this first part here, we see this tendency where things are basically going through this initial transient. Actually, the whole thing is really transient because there's no those fixed concentrations driving it. So there's that characteristic, and then we've got this part over here where we're going towards the what becomes steady state, because all the mass ends up going to C. So the question then becomes, how do I choose my folds? Now, one way to do this is I could choose, I mean, this is going to look very silly because nobody would probably do something like this. What you would, the sort of the knee-jerk reaction from the machine learning community is randomly choose them. But if you randomly choose your folds so that the reds are sort of, randomly spread apart, you could just have some bad luck where you have a lot more of the reds that lie in this region because you chose it randomly. I mean, over the long run, yeah, it'll be spread across the entire range, but you may just have bad luck with the ones you chose. And so if you want to get something that is a repeatable result where you see that the parameter estimates that you're getting and the measures of quality are consistent, you probably want to be more deliberate in your choice. And that's why something like this, where you sort of alternate over time, is probably going to give you something more reliable, because now the red, blue, and this sort of brownish color, they all have some of these early dynamics. I mean, actually, what we probably need here is actually more data, and that's actually a challenge as well. So if we had a lot more data, of course, much of this problem goes away, because then the random approach would probably work fine too. But if you have limited data, you do want to be careful to make sure you're covering all parts of the time course, and that's what this alternating approach attempts to do here. And actually, you may even want to consider maybe different alternating approaches too, different kinds of alternating. Right, that's what sort of the insight there is on the folds. In terms of cross-validation, really the main design choice you have is choosing your folds. That's really the main design. And there's a little second order details about what's your measure of quality. Is it R squared? Is it chi squared? Is it AIC? You have to decide on that as well. But I consider that really sort of second order. They'll all give you roughly the same thing. Okay.
We have a question in the chat.
Please.
So it says, no matter how you do it, wouldn't the data be not independent and thus make doing statistics with it much harder?
Great question. This is really one of the key insights from this particular technique. This actually comes from the statistics community. And you're absolutely right that the training sets are overlapping, but the test sets are not. And that's sort of the key observation, is that if you view the training as really sort of a deterministic operation, that you're going through a process of coming up with your estimates from a sort of deterministic set, where you get independence is on the test, because the test here is based on one set of data, if this is considered deterministic, and these are all on different data. And so I can't provide you with the details of the theory, but this is sort of a well-established technique in statistics where you do get, some kind of independence because of the separation of the test sets. And that's the reason why the folds have to be a partition. They have to completely cover the data because you want to use all the data, but they also have to be non-overlapping. Other questions? That was a good question.
I would perhaps have a follow-up question because I asked that question. And it was not exactly what I meant, but what I meant was when I split the time course, like is the second part, for example, not dependent on what happened in the first part? So I get that it is independent when you split it and only train on the train set and test on the test set, but the data itself is dependent on each other.
Yes, right. Yeah, absolutely. Right, right. Because these are time serial data. And that's certainly true. Anything that you want to do that analyzes, sort of errors at one time point compared to another. So, that kind of time series analysis, you have to deal with the fact that these are, that's a time course. And so there, clearly are time serial dependencies. The way this analysis is being done, though, is it's treating it as a batch rather than the time sequence. So, I've got this batch of data and I've constructed parameter estimates based on the batch. And now I'm comparing this to, and now I'm doing the predictions over here. And yes, you're right. The sequence, this is sequential data as well. And so errors at each point, you would expect to be correlated. Because if I'm off on one, there's an effect on the next. But again, it's being treated as a batch, and I think that's the perspective where you get independence.
Okay, thank you.
Sure, sure. Other questions?
Yes, there's another question in the chat. Is there a way to find out the optimal number of folds, and will we have the problem of overfitting?
Okay, so the optimal number of folds, I'm not aware, and this, my guess is that if you're really into statistics and statistics theory, this is probably sort of almost an analog to people who studied virtual memory in computing, and there are probably thousands of statistics PhD theses on that question. Unfortunately, I can't answer, the degree of success people have had with doing that. I'll tell you the practical approach that I've tended to use, which is I try different numbers of folds. And what you will, the original exercise that I had for this section, which I doubt that we'll actually get to, but I can at least point to it, was about evaluating for the Wolf model what happens as you vary the number of folds. So there are sort of two effects that are going to go on. One is as you increase the number of folds, so we have 3 folds here. So 3 folds means you estimate the parameters 3 times. And we've seen before, if you've, from our on Tuesday, that as you, each time you estimate the parameters, especially if there are a lot of parameters, like in the Wolf model, it takes some time. So the more folds, the more time it takes to estimate parameters. The other effect that you'll have, which you can see over here, is that if you get more and more folds, like over here, where we have a lot of folds, actually we have the same number of folds, so just chosen differently. But if you choose more folds, what will tend to happen is that each fold will contain the test set will have fewer data points in it, because that's what the folds are. You're dividing it up more and more finely. So you have fewer and fewer data sets. It tells you the number of folds is equal to the number of test sets. So that means the number of test sets, they may be chosen at points in time that widely vary. I mean, it could be, just this, little part over here or just a little bit over there. And depending on where you are in the time course, it may be harder or easier to make the calculation. The folds as themselves, the quality measure is about all of the points in the folds, how well they match. And with a larger test set, then the points which don't fit well are sort of masked by those that do. But with small test sets, you know, if I wanted to predict something that was like right around in here and that was the only thing I was predicting, probably won't do a very good job because there's a lot of dynamics going on there. So the net of this is that the quality measures and tend to vary significantly because you will see more variability in what you're testing against. The flip side is that the estimates of the parameters are probably going to be more consistent because you have larger training sets. You know, because the training set is what's left over after you take out the folds. You take out less, you have more training data. So you have much more overlap on the training sets. So that was a little bit long-winded, but that's the, you know, sort of the trade-offs there with the number of folds. And choosing optimally, I'm not aware of how that does. I mean, it's sort of this trade-off between computation time and sort of assessing some level of variability. A lot of times what I'll do is I'll try different numbers of folds just to get a feeling for where I am in terms of the nature of the dynamics in the data.
So just thinking about, like, your limits are basically, you could use half the data to test or to train and the other half to test, and then the other way around, and you have exactly 2 tests. And then the other limit is you drop one data point and run the thing and then test it against that one data point for all the data points you have. And are those, in theory, would they give you the same statistical power, or is actually the statistical power different for those 2 extremes?
Oh, I don't know how to, I haven't really thought about this in terms of statistical power. I guess it in part depends on what it is you're looking for. Are you looking at your estimate of the model quality, which would be, I'm using R squared, but the estimate of model quality is going to depend on the size of the test set, whereas the estimates of the parameters it's going to depend on the training set, and those are inversely related, right? Because if I have larger test sets, I have smaller training sets. No, I hadn't really thought about it quite that way.
Okay.
So I think that's sort of the way I would look at it. If you, and it's actually, it does point out that as you look at the results from cross-validation, be careful about what you're focusing on. Don't just focus on one or the other, just the model quality measure or the parameter estimates, their variability. You really need to look at both in combination because you're constrained. If you took the extreme case, like you said, of just, you know, leave out one, you know, if that's your approach to cross validation, you're going to end up with very consistent estimates of for the parameters. I mean, that's just because of the nature of data. It's like if you only did, if you left out nothing, obviously you always come up with the same answer. Yeah. So you're getting close to that. So that's why you sort of need to get a sense of both. And actually, you know, it's, you know, come to think of it, Lucy, you know, a reasonable thing to do is do both extremes and see how consistent they are. That might be an interesting experiment.
Yeah.
You know. Yeah. See because if I leave out a half and potentially even choosing different halves, you know, in different ways, that would be interesting.
Oh, like, different randomly chosen halves or something, or
exactly something like that. That might be another way to get at, you know, what is the underlying characteristic of my model estimates. Again, it's this attempt to try and make use of all the data when you don't have a lot of data. But then still get meaningful insights in terms of how good is my model, because you know, a very, very common mistake that people can make is they'll look at a measure of model quality and it looks good, but what's really being fit is the steady state or the end state of the system, and all the messy dynamics are sort of ignored because what they're looking at is data that maybe ran at, this is going up to time 30, maybe it goes up to time 300. You can imagine what's going to happen. This is all 0. These are all, you know, 50. I mean, that's a real easy model. Just say that, you know, C has all the mass and these have 0 mass. That's an easy model. And that's what you'd be fitting. You would be ignoring much of this.
You could probably weight your data in those cases, couldn't you? Like,
you would, right.
You would want to say, like, yeah.
And that's, you know, the reason why I can't, you know, really emphasize enough the importance of looking at your data. And I think that that's
right.
something that you really, and it's a challenging with biological data because it's very high dimensional spaces. So if you're dealing with gene expression, 5,000 genes is almost nothing in terms of gene expression data. So yeah, it's very, very challenging, and certainly any reasonable model in terms of the number of chemical species you might have present, it's very large. So, other questions? Okay, let me move on to the parts where we're going to talk about the computational aspect. So to give you an overview here, so this is, we're doing, I'm showing you design of experiments. We're doing cross-validation. So I assume you can see my screen. We're switching the browser window. I'm okay?
Yep, we're fine.
Okay, cool, great. Okay, so we just did the slides, and now I'm going to go on to the computational part, the cross-validation over here. And let's go up to the top. Now, one of the challenges with this particular notebook is, since it actually does cross-validation, there's some, computation time required because it's doing it with different folds. Also, this is another one where if you have not refreshed your notebook, I did make some changes. So you may want to quickly refresh and then just look at my slides or my screen while we're going through this. So I will run this. I think I ran this recently enough so that it shouldn't have to reinstall everything. Yeah, it looks like I'm not gonna, well, we'll see. Okay, I've organized the notebook in a similar way. We've got these preliminaries here that is just, all the setup stuff. You're welcome to look at it, but it's, the similar kinds of things. Yeah, it looks like right now what we're doing is we're doing the cross-validation. That's where the time's going to be spent. All right. So what I want to do in this notebook is go through some of the technical details with what's involved with cross-validation. Now, as before, the philosophy here is to give you a peek, sort of like, under the covers, what's going on. In practice, there are tools available, so you don't have to deal with a lot of the messy details that are involved here. But it's good to understand to some degree what's going on underneath so that you have a better feeling about how to interpret what comes out of a tool. So that's the intent here. Here's the model I'll use as sort of the running example. This is a linear pathway model. And then you've seen that one before. Here's the high-level algorithm that I use for cross-validation. And I sort of, again, this notion for those of you who are less exposure to programming and software, this is what we call pseudocode. It's not something that you could really run, but it gives you sort of like wherever we don't know exactly what we're doing, we just sort of say it in English. This is pretty close, but probably not exactly right. Okay, so we have this function called cross-validate, and cross-validate is going to do the cross-validation. It's going to, you give it a model and you give it some observed data and you tell it how many folds that you want. And again, we went through the details about what a fold is, this sort of partitioning of the data. Okay, so so the first step is we're going to construct the folds. So we've got this function here, which, the body of this notebook is really about, how do you write this kind of function? How do you do it? And so we'll get to that in a second, but we'll talk about the details for right now. It's sort of the overview. So we have something that generates folds. So to generate the folds, much like what we saw before in the slide deck, what we're doing is we're taking the observational data and dividing it up that many times. So just as a quick refresher, going back over here, that's this process here. We took this original data over here that is all in gray, and we put the colors on it. And this is, one way to construct 3 folds. So now we've got our folds, and then what we're going to do is, so a fold consists, I actually go over here. So you see, this is a fold right here. So a fold consists of data that are training data and test data. So each one of these are the 3 folds. And within each fold, we have a pair of training and test data. And I think this picture is helpful because the programming details can get a little bit confusing about what data structure you're actually dealing with as you go through this loop. So keep this picture in mind, and then we'll go down over here and see what's going on. So what this says is I've got folds, which is that collection of all of them, and I'm looking for each fold. So I'm going to pick a fold out of this set of folds, and in that fold, I'm going to have its training set and its test set. So we saw before. So like if this is the red fold, then the first training set. So if we're going through the set of folds, let's see here, let me show you. So this is, the concepts are, these are the set of folds, all these 3, and then I'm going to iterate across a fold. So this would be the first fold. And so in this first fold, here's my training set, here is my test set. So hopefully that becomes clear. Okay, so now I've got a training set and a test set. I use the training set to construct my estimate of parameter models, my parameter estimates from the model. So that's using fitting like with SB and then I have to evaluate the model using the test set. So again, I'll actually, probably I would have been better off saying over here if I put in the parameter estimates specifically here. Actually, I think I will, because I think that that's a little bit deceptive. I'm going to update this a little bit. This, again, this is pseudocode, so I can change anything. It's all really English. Estimates. Okay, I think that's going to make a lot more sense down here. Can I? Oh, I guess it's still executing. So that's interesting. Okay. Yeah, this is what's going on down here. If you're not familiar with it already from using your Jupyter notebooks or Colab notebooks, this gives you the status of where you are with execution. So what this says is I'm running something that's taken 5 minutes and 18 seconds. That's what's going on there. And this is cross-validation because you're doing this. This must be something that's being done, I think, on the Wolf model. It takes a lot longer. Okay, so back to the algorithm over here. So we got the parameter estimates from the training set. We're using the test set then with the test data. We've got the model, the parameter estimates, and then we've got the fold for the test. That's going to give our estimate of model quality. I typically use R squared, and then we keep track of the list of those model qualities. And what we're going to return then is a set of model qualities. The other thing that we can return, which I don't show here, and I probably should expand the algorithm some more to include that, is of course we're going to come up with separate estimates of the parameters. And that's of course, very useful as well. We'd like to see both the parameter estimates and the results of the evaluation of the model quality. All right, let me pause here for just a second because I think we're starting to transition where I think those folks who have less in the way of comfort with Python or with programming, there are going to be more details that it's going to be easy to get lost in the details. But I want to make sure that the high level over here makes sense. Any questions? Anything in chat?
Yeah, there was a question. Can you give examples of the tools, available for this process?
Oh, for this process. Well, there are a few different kinds of tools available. And first of all, and this may be a source of frustration for some, hopefully not too much, but actually, it turns out SB does cross-validation. And after you've done your fit, if you just say SB you get a fitter and fitter.cross-validate with the number of folds, that will do cross-validation for you. So you're going to argue, why are you spending this hour and a half when I could have just said cross-validate? So SB is certainly one tool that will do it for you and is intended to, fit into this context. I'm actually don't know if do you know if Kapazi does cross-validation? I'm not aware of that. I know they do.
I'm not aware of.
Yeah, I'm not aware of that.
I'm not aware of that either. Yeah, I don't know if they do cross-validation.
I don't think they do.
I'm sorry, Herbert?
I don't think they do.
Yeah, I don't think they do either. But there are the Python ecosystem, SciPy in particular, provides a lot of support for machine learning, and cross-validation is an integral part of machine learning. And so what you can do is get, for example, divide your data up. It will create folds for you, for example. And then it sort of roll your own then in terms of then you would have to go through the steps of, for each fold, you'd have to write your own loop. It would divide up the folds for you. Of course, you'd have to be a little bit careful about how it did that because you want to make sure you capture dynamics, the same dynamics in each fold. And that was the whole reason for this idea of alternating, in time. But assuming you can get it divided up into folds, then you would be responsible for writing this loop right here about constructing, doing a fit and then evaluating quality on the test set in each fold. I think that's what I'm aware of so far in terms of tools.
In the chat, someone else recommended scikit-learn, another tool for cross-validation.
Yeah. That's another part of the Python ecosystem. And again, this is sort of generally for machine learning. It's not something that integrates in Tellurium or things like that. What it would allow you to do again is some way probably to generate folds, but you would be responsible for writing this loop.
Are there things that you could tell it? You could give it the function that just estimates your data, like does the parameter estimation or something, like the parameter estimation function. You say like, this is my parameter estimation function. It will take in the data that you give it, and it will estimate the parameters. It will give you a result that is these parameter vector. Now go do the thing.
I'm not deeply. I would have to look at the details of what scikit-learn provides. It may be that it provides essentially a template for this loop with like callbacks.
Yeah.
So you would call something that much like the way optimization works in SciPy. You specify the estimation function, and then you'd also have to specify. Well, I guess it would divide things up into test and train and test folds. You'd also have to provide it with a function that does the prediction, because it's a complicated function. It's a simulation that does the prediction. So you'd have to provide it with both in order to then populate this loop here. It would be a non-trivial function to use, but it may be that someone has done something in those libraries, because clearly this is a common thing to want to do.
Yeah. They posted a link in the chat to scikit-learn's cross-validation module.
Okay, so cool.
You can learn more if you want. For more information.
Right, exactly. Right. Yeah, and again, it will provide you with a way of doing that, of doing these various parts, but you would still be responsible for writing the fitter and then writing the evaluate fold is something that you don't get out of the box from either obviously Tellurium or other, if you have a Copasi that will give you parameter estimates. So you could use Copasi to do the fit over here, but Copasi I don't know that Copasi provides you with a way, once you have fitted parameters, to do the predictions and calculate the quality of those predictions. I don't know if it provides that. I mean, Tellurium doesn't out of the box either. You have to do some, it's not a huge thing, but you have to do some of your own work to get there.
And I should say, this is one of the reasons why we provided Tellurium in the Python environment in the first place, is so that you can link out to other packages straightforwardly. We will give you data, we'll give you functions. If you need to do complicated things, then when there are things out there that'll do that, hopefully that's a seamless integration there.
Right.
Herbert also posted a paper.
Yeah, just an example I came across as cross-validation.
What was it about, Herbert?
It's a signaling pathway, I think. They do a lot of cross-validation, just an example. But then they use MATLAB, so I was hesitant whether to share this citation or not.
At least the technique should be good if people.
The technique, though, they go into quite a bit of discussion on the cross-validation.
Okay, great. Yeah, I'll be interested in looking at that as well. I mean, something might use as a reference for the class this fall.
Yeah, that's true.
Yeah, that would be very useful. Okay. Other questions on this loop? Because the next step is that I'm going to get into a little bit of detail about each one of these steps, so that I have, so you've got the loop over here. That's not too hard. Then the question is, so how do you generate folds? The fitting part, the only issue there is just, we already saw how to fit on day 2 in the afternoon, but you have to make sure you do the time alignment of the data, because now you've got a subset of data. You're generating a model over here, but what you're training but it has to then align with training data that's a subset, and it may have gaps in it. So you have to sort of cope with that issue. Evaluate the fold. So now it's comparing what you predict with some separate observational data. So those are really the 3 things that there's some details on: generating the fold, fitting the model, and then evaluating the fold. And that's the next step over here. So what I'm going to do on this next section over here, so I'm going to start with generating folds. I'm going to try and, again, as an attempt to try and be as inclusive as possible with this audience that has diverse backgrounds. And try and give you sort of the concepts behind it, the next level of detail. And I'm not going to dwell so much on a lot of the technical parts inside the functions I described, but anybody who has any questions, who's interested more in the technical details, I'm happy to respond to them, either during the time of this lecture, or afterwards if you have further questions. So let's talk about generating folds, and to reinforce again, I'm a strong believer in trying to provide some consistency and rigor when you write codes that you use for yourself, or actually you're going to be your own most frequent collaborator. It's going back to code you wrote 6 months ago is going to be your most common situation.
I like that analogy. You're your own collaborator.
So I think over time, and I find it extremely useful to have at least this minimal kind of documentation for any function that I write. What it does, what its inputs are, especially the data types and the outputs, and they have something in the way of tests. Tests also show you how to use it, as well as provide some confidence about the absence of errors, although obviously depends on the kind of tests you run. So what generate folds is going to do is it's going to take your observed data and divide it up into folds. And we know what a fold is. It's a partition of the original data and then within each partition, we partition it essentially by the test set. And the training set is what's left over after you take out the test set. I'll tell you, this over here is just to deal with the fact I may have different data types for the observed data. It could either be an array or a named time series. So, don't dwell on that one too much. The core of this has to do with how do we pick the folds themselves. So we have the number of folds is specified as an input. That's numFold. Okay, and so what we have to do is we have to come up with n folds. That means n pairs of test and training set. We have to figure out, we're going to focus on selecting the test set, because once we've got the test set, the training set is just what's left over after we take out the test set. So so the iteration obviously has to be across the number of folds, because that's what we're going to select on, as number of folds. And I call the indexing, this is going to be an integer. I call this remainder. And the reason why it's called remainder is that's the way we determine whether or not an observation in the data is going to be included in the test set. It's basically looking at the remainder when we do integer division. So n over here is going to be, indices is the index of the count of the number of observations. And if we divide by n by the number of folds, well, that remainder could be anywhere from 0 to the number of folds minus 1. And that's exactly the index over here. So this tells us the indices of the test set. And again, what I'm trying to do is alternate, one fold at the top. If I chose randomly, there would be a slightly different approach. So the test data are going to be the original observations at all those indices, and the training data is going to be the difference. And that's what this somewhat more complicated expression is about, using some of the set characteristics, the set functions inside of Python, saying that take the total set of indices and subtract from it the test set of indices. What you have left over are going to be the training indices, and then that you get the training data. So hopefully that is, at least the general idea makes sense about how you get these alternating folds. So this is this function, generate folds, and it, the input is going to be observed data. Number of folds in the output is going to be somewhat complicated because remember the data structure itself is complicated. It's going to provide you with a set of folds. Well, that means it's a set of, each element in that set is actually a pair of test and training data, and test and train, each one of these is an array. So that's sort of a mouthful, but that's part of the complexity here with dealing with this as well. So, but hopefully what this means is that in general, you just get to, if you use the function, you just get to see it directly. I've included a number of tests over here, both because I wanted to make sure it worked right, and also you get to see it working in different settings. All right, and I think that this stuff is already, actually my cross-validation finally finished. So that's good. All right, so that's the first. So what we've done right now is we've done this function right here. We've done right there. That's what we've done right now. So now I'm going to go on to the second one. Some of these are pretty easy. And then what this comes up with, of course, is what we're doing here is the iteration across the fold. A fold is a tuple consisting of an array of train and an array of test. So that we get automatic and get pretty straightforward. Now we're on to this next one over here about the parameter estimates. Estimating the parameters and evaluating folds. I guess I combine those together. Okay, let's take a look at estimating the parameters. Okay, so this is very much like what we've already done. This is not a huge adjustment from any sort of parameter estimation. We need the model. I include column names. We could have other arguments as well. The parameters to fit, that's just like what we saw before. You've got to list the parameters and ranges. And then we've got the training data that we're going to train on. The trick over here is we're specifying basically exactly what it is that would have been specified to SB Stoat already. There is another fitting argument, which actually, unfortunately, is hidden under the covers. I probably should have made it more specific. And that is you want to indicate how big the total data set is, so that you generate all the values. And let me show you the issue here. Okay, let's take this. Actually, this first one over here is a good example. So if I'm going to train my model on and produce fitted values on these data over here. So I'm going to train on this, and this is where I have actual data values, and that's where I can do my fits. And so I'm going to do my fits based on this. But when I come up with, when I want to construct a simulation to do all of the, fits, I have to include not just this time region over here, which would be just the early part. I want to include this part too, the red part, because that's where my test data are. So I have to be able to generate that as well. I have to specify where the end of the simulation is. And that's another argument that I think I pass as a separate parameter, which is not obvious there. But anyway, that's the only trick of any sort there. Okay. And then down here, we see tests. So it shows you how to use it. Let's look at this last part over here, the evaluate fold. So that's this one right down here, evaluate the fold. So now what we have to do is we have to construct estimates. So now we're not going to fit anything. We're going to run the simulation using the standard Tellurium capabilities of assigning the values of parameters based on some other source. So what we're passing here is this is the parameter estimates over here. And these are the test data that we want to compare against. So we have to go through and we have to construct the fitted data. So this is the simulation. We specify the end time. Actually, end time I think is a, okay, I fixed that one. That probably should have been done as a parameter over here. But you want to make sure that you have an end time that includes the entire simulation. That gets us our fitted data. And then we have the, this is where one of the challenges is that you're testing data. So, okay, so we have, here's the fitted data, here's the test data over here. And what we're picking up over here are the times associated with the fitted data and the test data, because it may be that your fitted data and test data don't come from a source that are totally compatible. So what this does is pick indices that are closest matching for each one. So you get them aligned right. That's the time alignment. Then we've got the fitted data, the fitted test data. So if you look at the fitted data and the arrays where we are going to compare with the test data, that's what this is over here. You've seen this before with, or this idea about, just laying out and computing the residuals. That's what's going on over here. We calculate the residuals for the differences between the fitted and the test data, and then you calculate the R-square. So a lot of this was about time alignment to make sure that when we get the fitted data, we're actually comparing with the right values of the test data. And that's where the sort of the technical details come in. Let me pause for a second, see if there are questions about this, because I went through it sort of quickly. It may be confusing. I saw something pop up in chat. Can you delete the outputs?
Define a time office.
Okay. Oh, okay. Oh, define.
That is actually a feature request that we got that feature request recently, and we were like, that would be a really good idea. So the answer is not right now, but if you follow that issue tracker thing, then when we do implement it, we'll let that know.
Hopefully it'll in the next few months, I hope.
Yeah.
Bias has been asking it for
I mean,
a while.
You can sort of get around it by telling Tellurium to only do exactly like from your first time step to your second. Say just simulate this and only collect 2 points of data or something, and then just do the next and then only collect 2 points of data, set the time appropriately and stuff. That's really awkward, and you don't want to. I mean, you could write a function that would do that, and it would make it a little bit less awkward. But yeah.
Coming soon,
hopefully. The other option, I mean, like if you tell Tellurium, if the original data is even, then you can just tell Tellurium to do that whole thing every time, and then if you have a subset of it, you can just match the ones that match. But it would be super helpful to have just like collect these time points, go. So
right, yeah, no, that would certainly. I mean. Right, it would certainly simplify what I have here. The way I do that, yeah, there's a little bit of overhead with doing the calculations. So. Let's see, I see a number of things here. Let me sort of go backward.
There was one more question to clarify. Fitted data is basically using parameters estimated from the previous step that the run Tellurium simulation is.
Exactly, that's exactly what it is, right. Yeah. So right, okay. And I saw something here about, reference to, apparently there's something, Uncle Bob with clean code about software engineering. So sounds good. I haven't seen that.
Oh yeah, I've heard of Uncle Bob.
Uncle Bob, okay.
Yeah. I've also posted a little past self making bad comments story.
Okay.
So.
All right. Okay, I think, did you see anything else in chat, Lucian?
I think that's it.
Okay, I think we're good for now. Okay, so let's go on to see where we are in our progress here. So we can generate folds, we can estimate parameters. Now we just did the fold quality, I believe. That was the last thing that, evaluate fold. Okay, so now we're on to the complete workflow. So this is my implementation, cross-validate. It probably needs to be scrutinized a little bit because I realized I had embedded some parts which were specific to this example. So I probably need a little bit of refinement here for at least in this context here. Yeah, so it goes through and it does the generation of the fold pretty much like the algorithm above. You get the parameter estimates, you evaluate the fold, and then you keep track of that, and then you get it at the end. Okay, so let's see here. If I guess what I should do is probably, let's do this. Let's actually run this guy. There's a certain amount of setup here that I will pull out. Yeah, okay, let me just do this and pull it out so you can see it in separately. This is with the linear model, so it should be relatively fast because running the linear model is really fast. So what I've got here, so these go, remember the parameter values go 1 through 4 corresponding to the K number. The observed data is our data from the time series. And so I'm going to use this function above, the cross-validate function, to do this. Doing differential evolution, you know, it probably doesn't matter too much. I probably can even, yeah, I think what I'm going to do is let's try this. I can comment this out. I think Python get very particular. So we can probably just, the default is going to be using gradient descent. And let's see, then let's see the answer in the end. Okay, how do we do? So it's doing, let's see, how many folds did I do? I said number, I guess I did 3 folds. Let's just out of curiosity, let's play with this a little bit. Let's do 4 folds. It'll go fast. It takes, obviously it takes a little bit longer because you're doing more time. So here we have 4 folds. You know, you don't see, you know, the parameter estimates are pretty consistent. I mean, to be fair, this is a real easy model to fit. So, you know, there's not huge surprises. R square of 0.8 or above is pretty decent. Let's see, I don't remember exactly how many points there are, but let's do 10 folds. Okay, and you can see it obviously takes longer. So now here we have 10 folds. This gives us a little bit more variation here. You see, you know, this goes back to the choose wisely, you know, drink from the right chalice. You can see some of them, you know, not so good, you know, probably pick things that were not so representative. You also beginning to see, this data frame doesn't actually calculate, but you're beginning to see more variability. If you look at the columns, look at the variance associated with the columns, you see a bit more variability. I think, can I go up to like 30?
There's more variability with more folds? Because I thought like the more folds you have, the more of the original data you're using. For
yeah, I'm sorry. The variability should be in the R squared. Yeah, this should actually be less variable. No, you're absolutely right. I was showing the wrong columns.
Okay.
Absolutely. Thank you. And avoid that confusion for everybody. So now again, I don't remember exactly how many points, so how many folds I can go up to. So I don't know how many I'm leaving out here. So now, you know, like what Lucian was saying, we should actually see less variability overall in the parameter estimates, but more variability in terms of the R squareds. And you can see it's clearly taking longer as well. That's sort of a qualitative insight. Here we go. But again, this is okay. So yeah, you know, okay, so take a look at that. Wow. You know, basically the first couple of decimal points pretty consistent across here with these columns, very much in line with what Lucian was pointing out. But let's take a look over here at the R squared values. I see here a 0.72, 0.88. We get below 0.7. I see a 0.97. So the range from basically 0.7 to 0.97, something like that. So, you know, it sort of follows the trend that we were expecting. Okay, let's see where we are here. So now on to the exercises. Actually, because the exercise will sort of be the break between the sessions, I'm going to come back to the exercise in just a second. I want to show you SB supports cross-validation directly. So what you can do is, if you look at this right up to here, so this up to here, Yeah, this is all right. Oh, actually, this whole section here. This is what you did on Tuesday. You know, you did, you gave it the model, the observed data, the parameters to fit. You got this fitter object. You said, okay, go ahead and fit. Then I get the, you know, give me a report and then plot it. And then this is the output of that. Here's the report, and then here's the plot over here. So if you took it one step further, that same fitter object, and just said cross-validate and say the number of folds, then it'll run it. And that's what actually was taking so much time. And then you can look at the R squareds that come out of that, and then the fitted values. So that would be another thing. And one of the observations here is we're seeing that I have 3 folds, and I don't remember, I think it was it 100? I don't remember exactly how many points there were in that data. But so that's, you know, it's a reasonable amount of data for each model. And so that's why you're that gets for each of the training, each of the test sets. So that's why you're seeing surprisingly high numbers here for the R squareds. But if you look at the standard deviation of, I was just fitting 3 parameters. The standard deviations here are not small. If you look at coefficient of variation, if you take the ratio of the standard deviation to the mean, I mean, that's about 0.3. That's pretty big. This is a little bit smaller. That's a bit better, but this one is not small. So, you know, the parameter estimates can actually, you know, if you had, you know, more folds and you have a larger training set, you probably get more consistency there. Okay, so that's just to let you know that there is an easy way to do this. Just, you know, once you did your SB Stoat setup already, just say cross-validate. You can go that way. All right, so here's the exercise. So use the Wolf model and fitting just these parameters over here, which actually I did down below with SB Stoat. Try some different levels of cross-validation. I actually, you may want to hold off on doing 100. I don't remember. It takes a little while, because remember, you're doing 100 fits on this data. Now, this is why I limited the number of parameters, so it wouldn't take quite so long. You want save this result and then look at some things. Look at what happens as you change the quality of, as you change the number of folds, at the variability in the parameter estimates and in the model, and then, you know, plot some distributions. It gives you more of a feeling, again, for something that's, you know, more characteristic of what a real model might look like. So that's the exercise. The solutions as, oh, actually what I did, yeah, just to let you know, this time I put the solutions in line. It's actually in this notebook, so I'll you know, you may want to close this unless you want hints. The hints will provide you with some steps along the way. So that's, if you need some hints, they're right there. Questions at this point, because I'm going to give you like, I'll say 20 minutes. We'll meet at 35 past the hour and reconvene, and then go on to discussion of design of experiments. Questions on cross-validation? I don't see anything more in the chat. Okay, all right, I'll see you in 20 minutes then. Sounds good. Okay, so I see in the chat there's a question about comparing uncertainties between, you know, different techniques for assessment. And you know, my sense is, of course, you have to be careful. I mean, there are 2 kinds of uncertainties. We have model uncertainty, we have parameter uncertainty. And you want to consider both of them. But in terms of comparing the measures, what you would expect is to see sort of like there's a correlation between the values, that, you know, if you're getting smaller R squares, you tend to get, you know, also larger chi-squares. You know, that would be the kind of thing you think, but I don't know, you know, that there's a direct translation from one to the other. It's more of a qualitative kind of thing. Okay, yes. Oh, okay, so some commented that like the SB Stoat runs folds in parallel. Unfortunately, you know, running in Colab, I think we only get one CPU, so I'm not sure that actually the parallelism helps that much. Let's see. Okay. So someone was getting an error at Tellurium. You got an error about no SBML lint object exists for J_K1. What that sounds like, I don't know how many folds you were running with. Oh, you found an error. Okay. But I will tell you this error, even though you found your error, it is still possible that that can happen if you have a larger number of folds. Especially if you have a larger number of, I'm sorry, a smaller, actually, it probably would happen with a smaller number of folds. So if you have less training data, that's where this is going to happen. If you have less training data, then sometimes what will happen is you cannot converge on the fits because you have less data to be able to work with. If that happens, then you actually will get no estimate for the parameters in at least one of the folds. And that could give you this error. So that can still happen. Other questions at this point? Let's see. Oh, okay, yeah. Unfortunately, yeah. Tellurium followed up that there was a typo because case matters in the specification of these constants. Yeah. Anything else that folks would like to discuss before we move on to design of experiments? Okay, let's move on to design of experiments. And let me make sure I find the right screen here. Okay, so we're done with cross-validation. And now we're going to do design of experiments. Okay, so design of experiments, this is very much has its origins statistics and constructing meaningful, how do you extract meaningful results from a set of experiments, of studies that you're doing to be able to understand what's going on in terms of especially some kind of science question. It really goes back to the notion of really another kind of modeling that takes place. If you've encountered the phrase factorial design, that comes from design of experiments. It's how you figure out, what combinations of parameters you should study. So you get to the point of design of experiments once you have a model that you have confidence in. So you've done model fitting, you've probably even done cross-validation. You have a sense that you have reasonable parameter estimates. Now you're going to use those parameter estimates in a series of studies to try and gain insight into situations that you don't necessarily have experimental data for, or maybe you have experimental data for those situations, but the data don't provide the detail that you want, and you're going to essentially expand the level of detail to gain insights into more what's going on with dynamics or particular the concentrations of proteins or metabolites that you cannot get directly from your experiments. So at this point, we assume that we have good values or reasonable values for the parameter data, for the parameter estimates. So the whole view, sort of like what's going on if we start talking about design of experiments, follows this flow. If you're asking a question, you've got your related work, some hypothesis, there's some sort of a model in here. So this test with an experiment, this involves a lot here, more than just going out and collecting the data. It's also the modeling part as well. And that's sort of where we are. We want to actually then be able to then figure out, what hypotheses we can construct and validate from our model. So the test with experiment, the experiments we're going to conduct are going to be computational studies. So there's a step in here in terms of the modeling that's not quite shown yet. Okay, so this is where design of experiments come in. Instead of going into the lab and doing something, setting up our glassware and then trying to, draw, actually measure something directly, we're going to, have done some studies up here that allow us to construct a model so that now we can vary some aspects of the simulation to then predict what the impact is going to be for a study. And just let me do a quick check. Hey, Lucian, I'm good in terms of sound and you can see my slides?
Yep.
Okay, great, thank you. Yeah, I have gone on for a long time sometimes without this. All right. Okay, so let me talk about the sort of the key core concepts here in experimental design. I'm giving as example here, I sort of stole some images from the internet, but thought they were pretty good to give some insights here. So there are three concepts that are really core to the side to experimental design and trying to be systematic about running studies. So the first thing is a factor. A factor is something you control. You're going to vary it. Now, in an experiment, in a lab, some things you might control would be temperature, how much starting material you have, maybe there's a catalyst involved. Those would be examples of things you can control in a wet lab. In a computational study, of course, we could control a lot more because it's all just going on inside of a computer, and we wrote the program, so we can control it. So there may be many other factors. You typically can't control, for example, the kinetic constant of an enzyme, but in a computational study, we can control the values that we set for kinetic constants. So we can speculate, gee, if this particular promoter is a stronger promoter than, that's reflected in kinetic constants, for, like a sigma factor, for example, or, the transcription factors, what is a dissociation constant? Well, it's a lower value, so it's, more likely to stay there and actually participate in transcription. So those are different kinds of factors. So then there's what's the concept of a level. A level is really the value associated with the factor. So the factor is sort of like the category, like, it's temperature or starting material. And then we could have different values that we assign. So it could be 80 or 100 in this particular case, presumably centigrade. So those would be two levels that we have over here. We have two levels for the material and two levels for the catalyst. And obviously the value of the level depends on the particular factor. And the response is what we measure. So the response is not something we control. We're observing this. Now, again, we're observing this computationally. It's not an issue so much with our measurement apparatus. So one way to look at this, if we just sort of focus on the factors and the levels, is we have this sort of cube in this case, it's in 3 space because we have 3 factors. In general, if you have n factors, it's an n space and probably not visualizable. But we have this notion of cube where we each point on each vertice in the cube represents a different combination of settings or levels. I'll use the term. A setting is the assignment of a level to each factor. Each point corresponds to a set of levels or settings for each factor. So for example, this point over here that I'm at right over there, this would be a combination of apparently I have, this is saying that parameter 1 is high, parameter 3 is low, and then presumably this one over here, parameter 2 is high. That's what this particular setting would be over there, and so on. So we have 2 values over here. So these would be the lows and highs. Every point here represents something different. And sometimes this is a common visualization. This would be if you want to consider all of these in combination, all possible combinations of this, then you have 2 possibilities here, 2 there, 2 there. So 2 times 2 times 2 is 8. So you would look at the 8 different possibilities in combination. And that's sort of the framework you work with design of experiments. Is you come up, you define the set of factors that you've got there. You're trying to choose how many levels, and then you also get to choose what combinations of levels, basically these vertices over here, you're gonna actually use. So, maybe you just decide to use a subset of these vertices because maybe you have a lot of factors and it gets to be, a lot of calculations to be able to, calculate on each one of these points, the vertices of the cube. So an experiment is going to define what level. So each point here corresponds to an experiment that was conducted. It's a settings, 2 factors. The levels were assigned to each factor. That's what an experiment is. An experimental design talks about which of the points you're going to do. So which experiments you're going to conduct. And then also what responses you're interested in. In this case, there are 2 responses. There may be many responses. So these are sort of the core concepts in design of experiments. So what we're going to do here, and which I find is a very useful way to think of things, is that we have, in terms of thinking about the levels, it's a lot of times very useful to think about some baseline, some nominal state that you start with. A lot of times someone will develop a model for a particular situation, like, a non-disease state that is, the starting point or the reference point. And that becomes the nominal or the starting values for each one of the factors. Those are, sort of like the 0th level. And so I refer to that as the baseline. That's a very useful thing to think about in terms of these kinds of studies. So, so that's a baseline. There's a baseline value for each factor. So that's sort of like, a particular level. It's the baseline level. So in terms of the Wolf model, what does this mean? So here's the output from the Wolf model using the assignment of to the parameters that was published in the model. So that would be our baseline, those initial values. Here are the various parameters. And so, yeah, okay, so here are the various parameters, and this would be the baseline. And then we're going to go and then start talking about what the responses are, the outputs are. In this case, the outputs that we've got are the values of the metabolites over time. Or there may be some other characteristic of the values of the metabolites. For example, it might be the frequency or the amplitude, the frequency that results when we say it, when we at a particular setting of the values of the kinetic constant. So the kinetic constants are all here in red. The responses are going to be the let's say a frequency that's associated with each metabolite. What is the frequency of these oscillations? That would be an example of a sort of a setup for an experiment in a study. Okay, now I'll warn you on this next slide and in subsequent slides, a lot of the design of experiment description involves a certain amount of notation. And I apologize for that, but it's sort of inevitable because we're talking about this idea of the combination of factors and levels for factors. And to describe that with precision does require some notation. So we'll go through this slide a little bit slowly just to make sure you feel comfortable with it. So y is going, the variable y will be the response. And again, much like, what we saw with the Wolf model, there are a lot of responses we're looking at. We're looking at, like a dozen or so metabolites. So I represents the particular metabolites, maybe I1 is glucose and I2 is fructose, 1,6-bisphosphate. This response occurs as a result. Oh, I'm sorry. I apologize. Y is okay, back up just a second. Y is the response, and I'm going to say for right now, let's just look at one metabolite. So let's look at glucose. Okay, the index I here is actually refers to a factor, not to the response index. So we're just looking at glucose here. If we wanted to look at fructose 1,6-bisphosphate, that would be a different Y. So this is just for glucose. Okay, so what we've done here is we've conducted an experiment. And in this experiment, factor I, let's say that is one of these guys over here. Let's say J1 _ K1, this parameter over here. So we conducted an experiment, and this factor J1, K1 had been set to a certain value. That's a value associated with this ith factor. Okay, if all factors, and that means that factor I is at this particular level, let's say it's 10% reduction from the baseline, and all other factors are at baseline when this experiment was conducted. So this is the output when that one factor, J1, K1, was set to, let's say, 10% of its initial value. That's the kth level for that factor. Okay. Now that's one experiment. So that's an experiment when we only changed one factor. Everything else is its baseline, and we've just changed that one factor. This is an experiment where we've changed two factors. So going back to the previous slide, then we changed J1, K1 and J2 underscore K. We changed both of these. So if we change both of these, we've got, I is J1, K1. This is, J2 underscore K2. Let's say both of them were a 10% reduction. That would be the particular K over there. And so that's the level for I, that's the level for K, and all of the factors are at baseline. So what we got, whenever you see this kind of notation here, where you see these two subscripts, an I and the case of I, you know that only one factor was changed. It was the ith factor, and it was set to this particular level. We could change the I factor. Instead of decreasing it by 10%, maybe we increase it by 10%. That would be a different case of I, a different experiment. Similarly, over here, we have two factors in combination. If there are, two different levels, we'd have to consider both of them separately, changing them. If suppose we have 10 different levels for each factor. So obviously over here, we'd have 10 experiments for each factor. If we have 10 factors, 10 levels, that's 100. Over here, if we have the same 10 levels, but we do them in combination, then we've got, this multiplicative effect. We've got, the 10 different levels for each factor. We have to consider them in combinations. So that becomes a much bigger deal. So here's some examples from the sort of, exercising, make sure that this notation makes sense. So here are the parameters. These are our factors, and here's the index of the parameter. So that corresponds to subscript. So a subscript when I equals 1, that's this one over here. If I equals 2, it's J1 underscore K1, and so on down the line. Here are the factors. So the factors I'm going to do in terms of a percent change. And this goes back to this concept of we've got a baseline. So obviously, a 0% change, you're at baseline. And then actually the 0% changes are not even shown. If you don't see a subscript for a parameter, like if I don't, like up here, let's say this was I, this was 1, case of 1, then that would refer to this factor being changed. And if it's case of 1, this is the first one over there. It's a -5% change. But you would know that all the other factors, all these down here, since they are not present, they have a 0% change. So they're at baseline. Okay, so here's some questions just to sort of exercise and make sure you understand what's going on here. So suppose I conduct an experiment and I get this response over here. And again, let's say, the response, we've got, about 10 or so metabolites. So you potentially get responses for all 10 of them. But let's say I'm just looking at glucose. So my Y reflects something about glucose. And let's say it's frequency of oscillation. That's what Y is, frequency of oscillation of glucose. So to get frequency of oscillation of glucose, I conducted, I was exploring this. I conducted an experiment and the index is 2, 2, 4, 5. Okay, what does that mean? So right away, I know since there are 4 here and I've got pairs of, factor and level, I know that 2 factors were changed. Okay, so which 2 factors were changed? Remember, our factors are parameters. Okay, so it's this second one over here. So it's this one here. I changed J1 underscore K1, and the change I made was the second change over here. So that's number 2. So it's 1, 2. So I decreased it by 2%. So J1 K1 was decreased by 2%, and 4, 5. What's going on there? So that's the 4th parameter, J1 underscore N, and I did it 5 was the level. So I increased it by 5%. and so on. You can see that over here, this one over here, and here's the answers. You can see, just to sort of test over here, if you see just two of them, obviously there's only one parameter that changes, and so it would actually be a simplification of this first experiment. Let me pause for a second because the notation can be a bit much, and I want to see if there are any questions. I don't see.
I have a quick question.
Go ahead.
If you have a percent change of 0, is that ever used? I mean, like y22 could also be y2233 or something.
Absolutely, right. You could, right. Yeah, you definitely could put that in there. That it would complicate the notation. This way, you know what is at baseline, but that would be an equivalent expression. For example, y22, comma 4, comma 3.
Right.
That would be equivalent to this.
Is there a use then for having your percent change of 0 on your list? If
you could,
wouldn't it just be assumed for everything
to,
not listed? Yeah.
Right. Yeah, you certainly could not include it. I find it useful just because it's explicit, but you definitely could eliminate that as well, because it's never going to appear as a subscript in your Y.
Right. Okay.
Okay. Other questions that you noticed, Lucian?
No, that was just my question. I didn't see anything else in the.
Oh,
in the
okay.
chat.
Okay, great. All right, good. All right, good. Okay, so again, the notation is probably the hardest part of this, but the notation is important so you're clear about what it is that you're doing. All right. Okay, so there are more complicated experiments you can do. Here we have 6 subscripts. So, if you have 6 subscripts, there are 3 factors, so you're varying 3 factors in combination. All right. That would be if these are the this would actually end up being what it is. You're changing the first factor by the first level, the second factor by the second level, the third factor by the fourth level. All right. Okay. So, and this is an example of what you just described. It includes the 3 over there. I mean, you can specify everything, but then this becomes more cumbersome computing in terms of notation. Okay, so now that we hopefully have that notation in mind, so if we have, let's say, if we have 3 factors, and we have an experiment that describes changes for all 3 factors. So presumably nothing is at baseline, or maybe the baseline is included in some of them, but not in all, in some cases, not always. So if we have 3 factors and we change all 3, that is what's called a factorial design. So, for those of you who've run into that before, that means you're changing everything in combination. There are good reasons. I mean, that will certainly show you all possible interactions, but it also gets to be somewhat complicated. We'll get to that in just a second. So the general way that this is described, when you talk about remember, part of what we're trying to do is, how do we do these computational studies? What things are we changing, to make what kinds of inferences? So if you change nothing, that's just your baseline. You haven't done any studies. You could change one thing at a time, one factor at a time, and consider all possible levels. So if we have, let's say we have 2 levels and we have 10 factors, we're going to do 20 experiments. So it's 2 for the first, plus 2 for the second, and so on, all the way for the 10th. If we have a 2-way design, and then we do all the experiments possible, then what we're going to do is we have to do the 1-way design, because that's actually getting us, this combination of we change a factor and then everything else is at baseline. So we still have to do the 1-way design, but then we do all things in combination. So, the first to the second, the first and the third, and so on. So we've got this combinatorics. We're choosing, the number of things we're choosing are the number of pairs. So it's sort of like, if there are n factors, we choose 2. And then for each one, we have, the product of the number of the number of levels that are possible. So that's the number of experiments that we have to conduct. And I think you folks saw that when you're doing cross-validation. And if you had more folds, then it takes a lot longer because you have to run, you had to do the, in that case, it was the parameter estimation was very time consuming. In this case, they're just simulations, which takes less time. But with the combinatorics in there, you get a lot more of them. So, and so on down. With three-way design, you do essentially, you have to do the baseline, and you have to do the one-way, and you do the two-way, because that takes care of the cases where it's 0. And then you get all the 3 in common. So now you've got how many factors you've got, choose 3. And then you've got the number of levels times the number of levels times the number of levels. So it's cubed. So this is one of the implications when you do a set of experiments. We decide basically your design decision right here is how many factors and am I going to vary in combination. So you can do 1 at a time, 2 at a time, 3 at a time. Those are the common types of decisions you might choices you might make. So this is a little bit, I'm not going to go through a lot of detail here given our time. It shows you about the combinations that you might consider. I wanted to move over to this one, counting the number of experiments in an n-way design. So this gives you sort of the combinations here in mathematical formula, where I gave it to you in words from before. It gives you sort of a feeling here. I didn't draw this out to some extent, just was trying to show the example, but the combinatorics from this, especially for a large number of factors like the number, these values of parameters, it can get to be big very quickly and also somewhat difficult to interpret in terms of what interactions really make a difference. And although the combinatorics are a consideration here in terms of the running time, the bigger challenge is interpreting what this means when you run these experiments in combination. And that's the part I want to focus on much more is what inferences can you draw? What are you learning from these studies? So let's take a slightly different look at this. And that is, if we look at a response, so this is a two-way design. I have two factors in combination at 2 different levels. And if I look, one way of sort of taking this apart, teasing this apart, is to say, okay, I've got this overall response here that I've gotten like a frequency. And how can I, what's my attribution about why I got this response? Well, one way to look at it is that there's some base that I'm going to get out of this, some mean value. Then I get some effect due to the ith factor and the level that it's at, and some effect due to the jth factor and the level it's at. And then there's something that's an interaction of those 2 effects. And the reason why this is helpful is that if it turns out that the I and J factors are really, essentially independent of one another, they're additive in this simple model, then what's sort of nice about this is then this interaction should be 0. In which case, I actually have learned quite a lot about what's going on in this system, because I know that these 2 factors can be varied, at least over the range of the studies I'm looking at. These 2 factors can be varied separately, and I get these incremental effects. And that greatly simplifies my insight into what's going on in the biochemical model, because I know there's separate factors. Got some baseline effect and then these incremental effects. If you are familiar with analysis of variance, this would be sort of like a deterministic analog for analysis of variance. I mean, the other thing is in reality, of course, you would have, replicas and some experimental, some variability, if you're especially doing something in a wet lab, and there'd be some sort of error term over here as well. I don't show that here because we're doing deterministic studies, but the same kind of analysis goes through if you're familiar with that. Let me pause here for just a second and see if there are any thoughts or comments at this point. Okay, I don't see anything.
So I've noticed that if you are including your zeros in there as one of your possible levels, you can save some time by skipping the reanalysis of that. Like if you're testing factors 1 and 2, one and you set 1 at level 5 at some change and factor 2 at no change, then that result can be used for when you test factors 1 and 3 is at no change, and 1 and 4 and 4
absolutely
and such.
Right.
So,
That's absolutely right. And you do get some efficiencies from doing that. Yeah.
so you can,
that's certainly right. Absolutely. Definitely a useful thing to do. Okay, so let me go on with this with the decomposition of the response. Okay, so the reason why I do it, I sort of hinted at this, is that you understand effects. The other thing that it allows you to do is that if your model proves to be effective, because you chose certain levels, like you chose maybe percent change of, let's say, 2, 5, 10, and 20. Suppose that's what your levels are. And I will primarily work in percents because it's units free, and then I don't have to worry about the specific units of the factors. So suppose I, basically went from, 2 to 20% changes in my factors. So now I want to make a prediction about, well, what if I make a 7% change in one factor and an 18% change in another factor? So I potentially then can use these alphas that I have at 2, 5, 10, 15, 20. If the interaction term is 0, then I could do some linear interpolation here and then come up with an estimated effect for what is it when I make these intermediate changes. And so that ability to predict is also a very powerful characteristic, but it's only possible if you see that the interaction term is modest, it's very small compared to these. And that's another reason why you want to go through this formality of this kind of decomposition and study, because I could just, a lot of times people will just do the interpolation. They'll do this experiment, do the interpolation, not go to the trouble of looking at the interaction term to see if that's valid, because that interaction term is going to, change the shape of the curve as I do different kinds of interpolations.
Could you also interpolate from that interaction term? Like if you've done a few of them?
You can, but essentially the interpolation is possible only if you go one level further, because I have to check to see if this interpolation term itself has, some interaction effect. So I'd want to go to a 3-way design before I did 2-way interpolation. I'd want to see that the 3-way interactions were 0 or close to 0, very small, before it's valid to do the 2-way the 2-way interpolation. Okay, let's
3-way. You mean a third parameter?
Well, yeah, right. So I would want to do, so this has, 4 subscripts. I want to do one with 6 subscripts. So I, J, and let's say M.
And why do you need that?
I need that because by doing interpolation, I'm assuming a linear effect. I'm assuming a linear effect. Okay. And I have to make sure that the only way I know that it's linear is that if I go to a 3-way interaction, I don't see some effect that's non-zero. It continues to just be a matter of the deviations from
even
the
if that
2-way.
Third factor is the same, even if you aren't changing that third factor?
Well, if it goes to even if I'm not, well, okay. If the third factor is constant, yes, that's true.
Okay.
But if I want, right, that's true. But if I want to be able to understand, right, if then essentially I have fewer factors if I'm not changing it.
Okay.
Okay. Let me see. I saw that there was something in chat because
will the process be affected if the factors are coupled? I think this is it. That's exactly what we're talking about. That beta
right.
term
If
is
the factors are coupled,
yeah.
right. And that's exactly what you capture with these interaction terms, is if the factors are coupled.
And someone says for the 3-way interactions, wouldn't need to check all the other factors and require all the 3-way interactions terms to be 0.
Exactly, right. You have to check all 3-way interactions, and you can see the combinatorics become significant, right? Because now you're talking about if you have n factors, it's n choose 3 instead of n choose 2. Okay, all right. So here's to hopefully reinforce these ideas, because again, I think the with cross-validation, the conceptual part was not hard. You're just dividing up the data. It was more of a challenge in terms of, some of the details of the calculations. With design of experiments, the conceptual part is a little bit more especially because it's complicated by this notation. Seeing so many subscripts, looks really, really busy. But it's the way we express with precision what it is that's going on. So here's a diagram that hopefully helps us. So suppose we have this response for a two-way design. So we're varying two factors, I and j, with their corresponding levels, case of I and case of j. And this rectangle is intended to indicate, the value for y. So this area really corresponds to the value for y. And what we're doing with this equation up here, this formula up here, is we're showing how this area got divided up. What corresponds to mu, to the alpha k, I ki, alpha j kj, and the beta. So here's how the area gets divided up. So first of all, there's the mu. That's this purple over here. So somehow, that takes up part of the area over there. And then next, we have alpha ki. So that's the effect due to alpha ki in isolation. And this is the effect of alpha kj in isolation. But the problem is that there's an interaction here where some of what alpha ki does is also done by alpha kj. Alpha I ki is also done by alpha j kj. Somehow they're both doing the same thing. That's what this overlap area over here is. And so that's beta. So obviously beta would have to be negative because we've got to, this is supposed to all add up to alpha I. This is all supposed to add up to the y. I apologize. Look at what I'm pointing to. I'm not speaking so well right now. This is all supposed to add up to the y sub I ki j kj. So if this is going to all add up, then if we have the overlap between alpha j and alpha I, then this part had better be negative because we've got to subtract out that effect. And that's what you would see. You would see a negative value here if these two in summation will then overestimate the y. Okay, so this is another chance to pause to make sure, because this is sort of intended to reinforce, but may bring up other questions in the process. I see something in chat. Okay, I see.
It was
if the factors are coupled, that's the same question from before. Okay, let me pause for a second. Anybody want to ask a question by voice? Okay, all right, great.
Can I
all right.
Sort of recap?
Please. Yeah, you recap, Lucien.
Here, so. All right, okay. So you are asking a question about what happens. So you're doing an experiment where you're varying two parameters, and then you want to say, like, for arbitrary levels of those parameters, what would they do, is your hope.
Right, you would like to be able to make that, so that is one goal, is you would like to actually be able to construct a model that allows you to predict the response for arbitrary values of the parameters with the constraint that you're making an assumption about some level of interaction is 0. So in this case, the level of interaction that we're hoping for 0 is going to be a two-way interaction. The parameters essentially are independent of one another.
So if all you are ever, if your only question about the model is changing those two parameters, then that's all you need. You don't need three-way interactions with parameters that you're never changing.
That's correct, right.
But if you want to say, like, I want to vary 5 parameters and then predict what the results of changing all 5 are, you're going to need to go interaction level until you start getting zeros, because you don't know. Actually, okay, go ahead.
Okay, so let me back up a second. So actually, with this model, if the beta is 0, then you actually could vary 5 at a time because you don't have to worry about interactions.
Right. So you have to go down as far as you need to get zeros.
Exactly.
As soon as you start getting zeros, then you don't need to worry about anymore.
Then you don't have to worry anymore. Right.
Okay.
And then that's exactly.
So if all you had was alphas and all the betas were 0, you're good, and you can vary all 5 at once. Right. If you start getting significant betas, then you're going to have to go to 3-way and hope that the 3-ways are 0. And if the 3-ways aren't 0, you're going to need to do 4-way interactions, etc. Until you stop getting correlated parameters, basically.
Exactly, right.
Okay.
Now, what you typically find, and what you'll see if you do the exercise, where actually maybe I'll just show you the results of the exercise, depending on our timing here, is that there are some interactions that are 0 you know, even early on, and the other interactions are not. So it's not that, you know, all interactions are zero, maybe most interactions are zero. And then what you need to do is deal with a subset of the parameters in combination.
Right. So if parameter 1 acts independently of everything else, parameter 2 acts independently of everything else, but parameters 3, 4, and 5 interact with each other constantly, then you just need to do the full experiment.
You need to do those in combination. And so, you know, you can see how it becomes increasingly complicated, but it also is giving you a lot of insight into the system you're studying about what affects what. So that's, you know, both of them go in combination. Let's see here, something in chat. I might be a little bit confused here, but what is the need for zero interactions? Oh, okay. So I guess, all right. So I guess that felt that that was answered. I'll repeat that anyway, because if one person asks it, others may be puzzled as well. So a zero interaction just provides you with a baseline. It tells you that's, you know, all the zeros is that's, you know, sort of like your starting point. It's a way of then describing sort of like what are the, by a default value for all of the other parameters. You know, in concept, I mean, obviously all parameters, and we're looking at parameters or factors as a more general term, all factors have to have a value assigned to them. By using the notion of a zero setting, the notation gets simplified a little bit because we tend to not do large number of n-way designs. It's, you know, going to be 1, 2, maybe 3. And so it simplifies our notation as opposed to having to have a, you know, pairs of subscripts for each factor. So that's what's intended. Okay, well, let me, oh, I see one more. Let me take this one other before I press on. Okay, how much is this different by looking at the correlations between responses for each factor? I mean, it's another way of getting at the same idea about what affects what. The difference here is by doing it in this way, you quantify the effect on the result for each factor. And correlation just shows that they're related. It doesn't quantify the degree of effect. This also, because it quantifies the effect, allows you to do the prediction and provides a way of sort of giving you a basis for understanding that prediction is reasonable. If you just do correlations, you won't get at this issue of, you know, multi-way interactions, which is a fundamental assumption, you know, the lack of multi-way interactions in order to do a linear extrapolation like this. And correlation is not a systematic way to do that. Okay. All right. So let me press on. Oh, all right. One more. This is one more. Okay.
That's right.
All right. What if factors 1 and 2 are proportional to each other? Then you'll always see interactions. Yes, you will. That's absolutely true. I mean, if you have factors that are correlated in that way, then you may want to reduce the number of factors. Actually, once you discover something like that, you probably would, you know, consider just only one of the two as factors. And that is something that certainly would be detectable from a correlation study. Absolutely. I'm not discouraging for you from doing correlation studies, and they certainly have value. I'm just saying that, you know, that if your goal is to actually be able to construct and isolate out the effect of different factors, this is a systematic approach. Okay, let me press on now. Okay, so then the next part about how do you calculate these values, values for the alphas and the betas, mu, alpha, and beta. Okay, so mu should be clear. That's an easy one to get. Mu you can get because it's just the baseline values for all of the factors. So that's easy. You just run a simulation once. And, you know, obviously if it's stochastic, then, you know, you have to run it more than once. The alphas are the deviations from the mean, basically. You know, what is the effect of this factor I at level k sub I? And so I'm just changing factor I. I'm just using one level here. What does that affect? Well, it's whatever I get as the response minus the mean. Well, the mean I got from the previous step. So that's pretty straightforward. So I just run simulations, you know, for each factor at each level in isolation, and I can do this calculation. And there's I'm sorry, I did this. I touched it too much. Okay. So you can see that there's sort of a recursive nature then for doing these calculations. Now, if I want to figure out the effect of, the beta, so now we've got two factors that we're changing. So I've got the result of running the experiment that's implied by the subscript of the beta. I subtract the mean and I subtract out the effect of the two factors in isolation. And now what's left over is the beta. And so on. I mean, obviously, the number of terms here is going to expand a lot because you have combinatorics just in the expressions. But that's the way you go about this. Okay. And again, I don't see anything yet. Lucian, was that somewhat clear to you? You're my test case. Yeah. Okay, great. All right, so that's how you do the calculation. All right, so here's an exercise. This is actually something that's sort of useful to think about, sort of like capturing these same ideas in a different way. So what I've got here is sort of a fictitious experiment. I've got, I should have labeled that over here. So this is actually the output over here. This would be Y, that first column. Here is the factors 1, 2, and 3. And the levels are just indicated by -1, 0, and 1. So here's the factors. And I'm not too worried about what their actual values are. Just, those are the different levels. Presumably -1 is low and 1 is high, and then 0 does have the interpretation of no change. So, and here's the results over here, the Y's. You can think of these as maybe everything is in units of percent. So this is, reduce it by 1%, and this would be increase it by 1%, and this is a 10% increase in the output. So here's something to think about for a second. Okay, so
wait. If it's a 10% increase in the, how come
in the output,
it's 10% increase for the 0, 0, 0 case? Wouldn't that by definition be 0?
Yeah, that should be over there. Oh, so I apologize. Yes, you're absolutely right. That's good. Yeah. Actually, let me fix that right now because that makes absolutely no sense. Oh, I can't. Okay. I will fix it later. Thank you very much for catching that, because that'd be a source of confusion. And that is a bug in the slide. Thank you very much. All right. So please ignore that. Don't pay no attention to the man behind the curtain. All right. Okay. So with that exception in mind, first of all, this is intended to be a two-way design. So that means we're varying two factors at a time. So keep that in mind. So as a result of this being a two-way design, why is it then that if you look across these rows over here, every row for the factors contains at least 1 0? Why would that be the case? Why is it that every row, there's no row where all three factors have a non-zero value? So anybody want to volunteer something in chat about that? Oh, I missed page. Here we go. Yeah. So the reason for that is that since it's a two-way design, we're only changing two factors. So one of the factors has to be the baseline, and that's why you get the 0. Okay, so the row with at the mean value, which one would that? Well, the row at the mean value is the one where we don't vary anything, and that should be this over here. And then I think Lucian, I think the issue was that I wasn't doing these in terms of percents. I guess this was supposed to be my baseline value. So I think that's really the correction I need to make, is to say that these are not percents. This is the baseline.
They're actual
value.
Like outputs then.
So these are actual
at the
outputs.
baseline, the output is 10.
Right. At the baseline, the output is 10. So the baseline here is 10. The other thing you can look at is you can look at if you want to see the cases where it's essentially, if you wanted to just pick out those rows that correspond to a one-way design, which rows would you look at? Well, you'd look at the rows where only one of the factors has changed. So for example, let's see here. Here's a row where only one factor changed. So that would be something in a one-way design. Here's a row where only one factor has changed. Let me see here. Trying to get the reaction.
Third row is like that, yeah.
Yeah, two factors, zeros. Yes, thank you. Someone answered it for me. That's another way of putting it. Two of the factors are 0. That's exactly the same thing. And similarly, if the rows that correspond to the three factors or the two factor changes have two of them changed. All right. So
can I
hopefully
ask
this is
sort of backup? I have sort of a backup question. It's when you say the output is 10.
Yes.
In a model, like, is this like the steady state or something? And you look at a particular output at the steady state, and that's why you only have a single value. And
yeah. I mean, that's a great question, right? And so this is a question of what is your response? What are you measuring? So I've been doing this all as scalars values. So maybe 10 is the number of, the estimate of the frequency of oscillation of a metabolite.
Okay.
That would be a scalar. But, it could be something more involved. It could be,
like you could have a time course. And
you could have a time course, right?
All of the plots, all of the points in the time course or something, yeah.
So let's think about that, at least as an experiment to sort of, get these concepts down. If we go back here and we look at this, just more focus on just this equation here. This is what I want to focus on. Suppose now that the results of an experiment, the y, it's no longer a scalar, it's a vector, all right? So what does that mean? Y is a vector. That means that all of these had better be vectors, because otherwise it doesn't add up, right? So that means that, each one of these effects, so when I compute an effect, we're talking about a vector of an effect. So that it adds a different dimension of complexity to it, but you have to be thoughtful about what you mean. Another way to do this, which is a little bit of a cheat, is, essentially, for like the wolf study, our response actually is a vector, even if we're looking at frequency, because we have 10 metabolites. So we really have a vector of frequencies because we have one frequency for each metabolite. So already we have a vector, but that means then essentially what we've done is we've done 10 different studies, one for each of the metabolites, and we've come up with 10 different values for the alphas and the betas and the mu would always, actually the mu's are different because they're different outputs. So that's it's just another part, in terms of, again, it's all about thinking about the notation, being clear about what it is you're measuring and you're studying. All right, so hopefully with these various paths through, the core concepts make sense. Let me see if I have a couple more thoughts here on the slides. Yeah, so here's some other things that you can use. You could, you might have replicas. You might have, do something with, maybe you're going to do doing some cross-validation with it. Also quantifying stochastics, and then also you might want to, you've got the complexity of this design itself, because with if n is larger, then you have more things you have to figure out in combination. And we alluded to this a little bit because we made the comment that if you do a two-way design, you may find that most of the interactions are zero, except for just a few, maybe like there are 2 or 3 that have some interactions. And what you probably would want to do is drill down on those particular factors and do a higher level design, maybe n is 3, possibly even 4, to see what level of interaction needs to be considered to actually get the beta down to 0. All right, so that's what I had on this side, so the theory part. Let me quickly, we have a half an hour left. So what we're going to do here is we're going to go over to this notebook, Design of Experiments. Let me do the run here. Get the notebook back up. I think what I have time for here is to give you sort of a high level about what's involved with running the experiments, setting this up, and the computation part. I'm not aware that there's much in the way of support. I know that SciPy and it may be scikit-learn, I'm sorry, does provide something in the way of factorial design, but this, and something I believe in terms of analysis of variance, but that's really from the framework of doing a study of observational data, not this idea of doing this kind of simulation study. So I don't know there's an out-of-box solution to applying these ideas directly. There's a little bit of work you probably have to do to put together the various codes to be able to do these studies. So let me give you sort of an overview about what's involved to do this computationally. So obviously you have to be able to run a simulation with factors at designated levels. So you'd want some code that says like run simulation with this kinetic constant at this value, this kinetic constant at that value, and so on. You've seen me do this a few times where I'll pass a dictionary to the simulation code, and from that dictionary it picks out the individual constants in the model that are set to specific values. So that's the way you do this first step here, is being able to have that kind of function available to you. Obviously, then you want to be able to calculate the responses from the simulation results. And the non-trivial part of this is part of the discussion Lucy and I were having. You run the simulation and you get a time course coming out for various like concentrations. So you may see a time course for glucose. But that may not be your response. The response you may want is actually the oscillation frequency of glucose. So that doesn't come out from the simulation data. There's some separate analysis of that. You might use a fast Fourier transform, for example. And there may be some considerations there that you want the oscillation at steady state. So, or after, at least after the initial transient, there's a question as to whether there's a steady state there. But after the initial transient is where you want to do that. So you may have to have some cutoff point that I've passed the initial transient and now I should see the oscillations. So it's a little bit of work there then sometimes to calculate what your response is actually going to be. After you've done that, now you have this capability. Now you want to actually run the experiment, construct a table of responses for the y's at each of the factor levels. So now I've got some sort of a loop that's going to vary the levels of all the factors, compute the response, run the simulation, compute the responses, and then construct a table that shows all of them. And then I actually want to sort of, now I've got a table of responses. These are y's. From this, I want to calculate the alphas, betas, whatever else I'm doing, gammas, how many level of interaction I'm doing. From this, I actually have to calculate those levels. So that's sort of the game plan overall. Let me give you a flavor of this. This one here I'll touch on briefly. This one, probably a little less briefly, but then I'll focus more on these two. I saw there was something.
Yeah, it says, aren't the mu and alpha values coming from a specific simulation?
Yeah, right. Each one of the mu and alpha values are coming, right, but we're gonna have a collection of them. So so there will be you'll run one simulation to get mu because that's just the baseline. The alpha will depend on the levels of each individual factor. So 10 factors at 2 levels, I'll have to run 20 simulations because I have to, for each factor, I have to consider all the other ones will be at 0 except for one factor. So I'll run 20 simulations and so on. Hopefully that clears that up. Okay, so these are usual preliminaries. So I'm going to do this example on the Wolf model. We've seen this lots of times before. So my factors are going to be these constants over here, considering the different levels. All right, so this is the model. I believe this is with the, this is with the synthetic observational data. Okay, so then let's talk about what our study is, because that's usually the whole motivation for this effort was that we want to investigate some science question. And so, let's start with what the question is. So the question I posed here was, I'd really like to understand what factors affect these oscillations. What are these oscillations really sensitive to in terms of the settings of those parameters? And so, the design itself is then going to have the levels, that the factors are going to be the kinetic constants. The levels are going to be percent changes in the kinetic constants from baseline to see how much variation in which levels I have to do in order to change the oscillation frequency. So that means I have to, that gets on the next one is what are the responses? Well, the responses are going to be frequency for a metabolite. So each metabolite would have its frequency. I'm going to focus on glucose just so that I only have one value to look at. So I'm looking at the frequency of oscillation of glucose after the initial transient. That's going to be my response. So that means I have to calculate something that gives me that. I use the fast Fourier transform. I'm not going to have time to go into any details about how this is calculated. You'll just know that this is a measure of frequency. So I said one of the things that you need to do computationally is you need to have the capability to run a simulation so that you can change parameter values. So here's some code. I'm not going to go through much detail other than just describing how it's called, what's called the signature, what you have to provide and what gives you back. So you name a parameter like J1 underscore K1. You tell it a set of percents you'd like to change. And then this is, whether or not you get a plot when you come out of it. And this works for the Wolf model is what's going on here. And so here's an example of using it. So this is run simulation where the percent changes. I think this goes from 2 to, I guess it shows it up here. Oops, go back up here. You see the percent changes. It goes from 0 to 10%. And you can see what happens as we change. This is just changing J1 underscore KI. So this is baseline. This is exactly what you get from the Wolf model. If I change it by 2%, so I'm increasing J1 under KI by 2%, 4%, 6%. I increase it by 8%, I have extinguished the oscillations. Now, the output that this is showing all the simulation values. Of course, what I want to do is I want to have a way of quantifying. I want to change this into a scalar, which is the frequency of the oscillations for glucose after the transient. And that's what this next part is, the calculating response. So what this does is it says, well, this is calculating the fast Fourier transform. I'm not going to go into detail about that one. This is plotting it. This is what you see when you plot a fast Fourier transform. You show a magnitude at different frequencies. So this is what you're getting over there. And what this is showing is pick out the peak frequency. So again, we're calculating response over here. And so that's actually, let's do this calculation here. Let's show you what the value is. So if I say calculate peak frequency for glucose, this is just for the test to make sure it's working right. Yep. Okay, so if I actually do that calculation, calculate peak frequency using the Wolf data, and it says the peak frequency I get is 0.05 for the particular baseline parameters I was getting. Okay, this is doing this for it's calculating all the responses, the Y's, for a one-way design. Okay, so now we're actually getting to the point where we're calculating, this is that, I think it was the third step in the design. So to do a one-way design, we consider each factor in isolation. So it says here are my factors. And let's see, let's show you what the factors are. So if I look at Wolf factors, take a look at that. What it is it's the names of all the parameters. So those are my factors. And I'm considering what percentage changes. Well, the default is I'm not changing them at all. It's 0. That's just for default setting. In the test code over here, it considers a minus 2% change and a positive 2% change. So let's see how the code is implemented. So it's a one-way design. That means I change one factor in isolation. And so what goes on here is it's running the simulations with each factor in turn, changing just that factor by the set of percents that it was indicated there. And then it accumulates what those changes are in terms of the percent changes. It has to calculate the frequency. So it uses the result, the that peak frequency calculation. So it passes on, here's the percent change, here's the molecule that's changing, and keeps track of, for that percent frequency, for that percent change, what the frequency was that resulted from that. So that may take a little bit of looking at, but here's the end result. What you get a table. This is a data frame. The rows over here are the names of the different parameters, and the columns are the percent changes. So we're considering from a -20 to a +20% change. And so you see that, for example, if we go down this column right here, you notice they're all the same, and that's because nothing has changed. That's the 0th level across all the factors.
So if you wanted to be a little bit more efficient,
you
might just not run it 20 times because you're actually running the same simulation 20 times here.
Exactly. And that's a really good point. That would be definitely an efficiency improvement, right? You'd only need to run this one once, and then you could just replicate it. And then across any row, you see what happens as you go to the left or to the right in terms of increasing or decreasing. Now, these are the Y's. What we're really interested in is we know, and actually from this, of course, you get the mean. That's going to be just this column, which is all the same. That's the mean value. But we would like to see the alphas, because then that'll get a sense of what the effects are relative to the effects. And that's what this one over here does, calculate the mean, which is pretty straightforward, and then the alphas, which is a subtraction from the response value. And that's what this table gives. It gives the alphas, and you see that some of the parameters have an effect and some of them don't. So for example, and sometimes the effect is not at all obvious either. So for example, you certainly see a larger magnitude effect with J1 underscore N, as its magnitude of change is larger. So, either plus or minus. A minus change seems to have more impact. But what's also interesting here is that you're seeing, at least over here, this is very curious, is you see that the frequency increased by 0.3 and then it ended up decreasing, almost plateauing down here. So I would want to go back and understand that better. That is not, I'm really surprised there's some sort of thresholding effect that's going on there. What's going on? And also the fact this is positive and then we went negative. I would really be curious to understand why that is. This over here on this side, it makes more sense.
Coefficient?
I believe so. This is a Hill coefficient.
Yeah,
right.
I think if you put that lower, then if you put that too low, then there is no Hill coefficient.
Yeah, then there's no Hill. That's
right?
true. That's probably, yeah, that's a good point.
I think.
Yeah. Actually, you know what? It probably went to 0.
Yeah.
It could very well, the Hill coefficient could have gone to 0, and obviously it stays there. So yeah, you're probably right. That's probably, because that's the Hill coefficient. Yeah, I think you're right. Some of these others that have no effect, some others, like a K. So yeah, that's a really good point, Lucian. So the Ks are the constants, and those have a more reasonable interpretation. Lower values tend to be on one side of the effect of on frequency, and higher values are on the other side. So this one, a lower value of this K decreases frequency, and larger value increases frequency. I think we have the opposite effect on the influx here.
What about J1KI?
I was just looking at that one. Yeah, I can't explain that. Yeah, that's another one where we have some sort of thresholding, don't we? I mean, the fact that we end up with a -4.8 and a -4.8, I think we're hitting some sort of boundary there. Yep.
Yeah, that seems to be the point at which you don't have any oscillations anymore.
Exactly.
Oscillations have been killed.
Yeah. So yes, that's why we get the threshold, is because that's 0 essentially. That's no oscillation. Because we're at 5.1. Once we go down to 0, we're at 0. We're not going to go below 0. Yeah, I think you're right. I think it has to do with the characteristic of what Y means. Yep.
I have another question here. So some of these things don't affect anything at all, like J4KG or something. But so what we were saying earlier, that once you get a 0, you don't need to worry about it again. But it occurs, but I'm guessing there may actually be an interaction between J4KG and J1N, say, like if J1N goes really high, then suddenly J4KG changes actually.
Some kind of a thresholding effect, right? So in this, right now what we've presented is actually incomplete because we have not shown the two-way interactions.
Right.
This is just one way.
Yeah. These are just the one-way interactions. But I was just remembering that we said, like, after you get two-way interactions with zeros, then you don't need to worry about three-way interactions. And here we sort of have one-way interactions at zero, so you don't need to worry about the two-way interactions, except I think you do.
Like, no, you can't. Right. The way to interpret this is if you're at an n design, so the n is 1 here. In order to say that, you know, I believe this, I have to go to n plus 1. So I can't really believe that that's really true until I see at n plus 1 that there's no interaction, that there's no n plus 1 way interaction. So since we're only at n, yeah.
I mean, even then, though, I can imagine situations where n plus 2 you suddenly have interactions, like, right?
Oh,
like,
okay, with more combinations.
Even with more combinations, right? Like J4KP doesn't seem to affect the oscillations,
right?
Except maybe it does when in a completely different
in some edge case. So, well, that's true. You are, yeah, and that's true. That's true. I mean, that is possible. I think the
just gets less and less likely, I guess.
Yeah, I think that's sort of the argument there.
Okay.
Right.
All right.
Okay. So let's see. We're at a quarter till right now, and I'll go over what the exercise is, and maybe it probably is most productive if we actually just sort of look at the exercise. So what we're doing is we'll compare the effect of parameters on glucose and with okay, so we're comparing how the parameters affect glucose versus how they affect NADH. That's the idea. So we have the same set of parameters, obviously, for both. You know, do we somehow affect the frequency of glucose oscillations in a different way from NADH oscillations? And are there some parameters that have more of an effect on glucose versus NADH? Because what we're doing here is we're just looking at glucose. And the fact we see all these zeros here doesn't mean that they're going to be zero for NADH. Okay, so let's take a look at this. So this was just a matter of running the studies for both glucose and NADH. And again, it's not really the full investigation hasn't been done because we didn't consider the two-way interactions to really interpret this fairly. And what we're interested in, but assuming that had been done and we saw no significant two-way interactions, one way to be able to look at this is to say, okay, I ran it for glucose, I ran it for NADH, I got out of this the alphas because I'm primarily interested in those effects. And what I'm interested in is there something that affects glucose more than affects NADH. So if I just look at the difference in their alphas, that would tell me something. And this is actually sort of interesting because we don't see a difference in most cases, except for this one cell up here. And that's sort of interesting in terms of the input driving. Everything else, looks like it's the same. But that is different, and we do begin to see an effect. So it might be interesting to expand the range.
J8 and J8K1.
Oh, I'm sorry. Was there another one that I saw? Oh, you're right. I didn't even see that one. I was blind. Yeah, good point. That's a problem with the table. Yeah, this would definitely merit some investigation, right? Because it seems like there's some kind of interaction. There may be some kind of interaction effect because why is this 0.3, but this is 0, and this is more extreme? So I would immediately be suspicious about what's going on there. Yeah, and that may have to do with some, there are these thresholding effects because, as we decrease the kinetic constant, exactly, right. We discussed this earlier. My guess is that what's going on is that there was an effect here, but as we go to a -20 on the differences for both of them have reached their threshold, and that's probably what's going on. But we would want to investigate that.
Can you scroll up and look at one of the J8K1 rows just to
up here on?
Yeah, there we go. J8K1.
Yep.
Oh, it only gets a -0.3.
Yeah, but we also would need, this is glucose. We'd have to look at, oh, I'm sorry, this is NADH. Yeah, right. This is NADH. That's -3. Then we want to look at glucose. Did I do? I thought,
Is that what
here's
we did
glucose,
earlier? Yeah.
Yeah, I think this is glucose, right?
Oh yeah, so there it is. That's the J8K1.
Right,
So
right, right. So you see
J8K1
0.3
goes
and then there's a 0 over here. Or, I'm sorry, -3 and then the 0 over here.
right.
Actually, it's
And so,
interesting that, go ahead, you finish.
so that's it. So at -10, for if we're looking at glucose, it takes
Right.
-20 to make it drop down to -0.3. But for
Exactly,
NADH, it only takes -10 to drop down to -0.3. But after that, it stays. So
It stays, right, right. So maybe we want to expand the range a little bit to make sure that we have a good understanding of that, or even get finer gradations of changes over here too, to make sure we understand that fully. Okay.
0.3 seems to be the unit of change also. Like we see
Yes,
0.3,
right,
0.6, 1.2. It's like 1 unit of any change. That's the smallest amount we get.
Right. Yeah,
I
and
don't know
that's
why,
interesting
but
because I didn't have, right, that is a curiosity, right? And especially since these are not even percents either. I guess these are, 5, 10, well, sort of doubling over there.
I think it's more in the responses. Like
Yeah.
it doesn't matter what your change is, the measurement of your response is just goes by 0.3s.
Right, right.
Everything is
Yeah.
seems to be a multiple of 0.3.
Yeah, and that's an interesting one too, because then you get into some concerns about, well, gee, maybe do I have enough points here? Because I may be limiting the granularity of my frequency that I can detect because I have too few points. So I may want to go back and do the simulation with more points, because then maybe I might be, finer granularity in terms of detecting frequencies. Okay, hopefully all this discussion was helpful. I see one response in something in chat.
Yeah, Polly was saying it might have something to do with
But
the
it had,
fast
yes,
Fourier
exactly.
transform.
Yeah, exactly, Polly.
I
That's
think
exactly what we're
exactly
saying.
Right.
Is yeah, we may need more points because that may go back to the FFT. Okay. All right. So we only have a few minutes left. What I would suggest at this point, let me see if there are additional questions that I can answer. I tried to, let me sort of review, actually what I will do is I'll review the algorithm up here that you have to go through, and this is something that, maybe we need a tool or some help so that folks can more readily do this kind of thing. I think design of experiments is not something that has really made its way into the computational biology community in terms of, how you do computational studies, but maybe that's something we could promote with appropriate support. So again, the kinds of things you have to do, you have to be able to simulate with factors at different levels. You're going to get out, time course values of the concentrations. From that, you want to calculate what your responses are. In this case, we're using frequency. And then you've got, a table that will show the values of responses at the different levels. And from that, you want to calculate these components of response, the mu, alpha, you might have a beta as well. Questions at this point? The comments on chat. Okay. Oh, we have an eager person here. General question. Let's see about this approach. Oh, okay. So general question about the approach. How different is it from running simulations by running these parameters at many different values? So it's a thought like, like randomly choosing values of parameters to explore the parameter space. So go ahead, Polly, you're going to say something? Yeah, go ahead.
Yeah, sort of like there are these people who really try to find like almost a very dense set in parameter space and say what kind of dynamics can you expect? Like how different is this approach from what they do?
So in theory, this should be much more systematic. Because what you're doing is you're systematically looking at how parameters affect one another. It really has most credibility for interpolation rather than extrapolation. So you wouldn't want to then make a statement about a level that is beyond what it is that you've evaluated. So if you've gone, from -20 to +20 in terms of percent changes, you wouldn't want to make a prediction about +50. But if you're within that range, then it's a systematic approach. And the fact that when you consider the when you're looking at the interactions going, if you're going to be making a statement about an n-way design, and you've done the n+1 way design, so you see low interactions, you have a strong basis for saying, I understand what it is that this will actually be. If you randomly choose them, you're exploring the space at those points. But if you're trying to make a statement about, what are the effects of the different parameters, you really don't have a strong basis, because you've sort of confounded it, because the parameters have to be considered in combination.
I mean, as a related question, even this example that we went through, we're seeing that like when there's a bifurcation, sort of like what the interpolation breaks down a little bit. So can we really say that if the response are somehow monotone, then you could interpolate sort of safely? But if we're seeing kind of change in monotonicity, or like the response is increasing then decreasing, then we need to fine grain it and restrict ourselves to like the region of the parameters where we can safely interpolate. Is that correct?
Well, what I would say is, yes, you're right. I think what happens is that we focused on the one-way design, which is definitely making the assumption of monotonicity, not even monotonicity. I mean, it's even saying linear. So you definitely want to go to higher way designs if you expect a more complex surface. And what you were describing, I think, is a good way to do it. Drill down on that section where in this region, it does appear to be linear, because that's really the strength of where this design of experiments comes in, is if you can drill down to an area where the responses are actually linear. That's when it'll be most effective.
Thank you.
Yeah, sure. Thank you. Okay, so yeah, I guess the chat was all from you, Polly. Okay, other comments or questions? Okay, and feel free to contact me afterwards if there are other questions that occur. I'll do my best to respond, see if that's helpful. So I think at this point, yeah, we're pretty much at time. So you will have other things. I guess you're talking about containers this afternoon, containerizations, Veronica?
Standardized formats and reproducibility.
Okay, standardized formats and reproducibility. Okay, very good. All right. Last chance.
Veronica, do you have any, is there going to be material on the drive for that, or is it was blank earlier?
I'll upload it soon. I'm just fixing an error.
Okay, great. I just wanted to know what people should expect. So great.
And there's also something about other tools or not?
Oh yeah, and we're gonna do a transition period. So
yeah. Okay, great.
So transition is about going from the class to back to their work environment to try and help them incorporate the what they've learned.
Well, tooling. What tooling is available in terms of Python IDEs?
Mm-hmm. So
okay,
we can all kind of share the environments that we work in.
Yeah.
A little bit,
okay, sounds great.
Yeah.
All right. Well, thank you all very much. Enjoy the rest of your day or evening wherever you are.
Mm-hmm.
All right. Take care.
Thanks.
Bye.